Hello. So the second portion of the chapter on the digestive system is the physiology of the digestive system and the process of digestion. Before getting into digestion entirely, there were just a couple of auxiliary points which are made. First, we obviously have to control the um, the digestive system. And so the autonomic nervous system plays a great role. When we're under stress, we reserve our body's resources and blood for the muscles, for the heart, for the skin, for those portions which we use if we're going to run or fight or need to cool down as we sweat, as we run, etc. And so the sympathetic division largely will direct blood and resources away from the digestive system. It is only the parasympathetic division which fully allows the digestive system to function at its best. In addition, then there are other, oh, and I'm sorry. And for that reason, for example, if you were under stress, like before you were to give a public speech, your mouth would become dry because under the stress, you're not making as much saliva as you direct blood away from salivary. Um, in addition, there are a couple of other things to consider for the stomach. First, the head controls the stomach. So when we think of food, when we see food, taste food, smell food, that can stimulate the stomach. Secondly, the stomach can stimulate the stomach. The stomach has its own nervous system, that enteric nervous system. It can detect stretch. It can detect pH, acidity. It can detect the presence of peptides. And so therefore, when the stomach is stretched, when there's food in it and the acid level goes down, when there are proteins in the stomach, the presence of peptides, this stimulates the stomach. The stomach can stimulate itself with the hormone gastrin made by those green cells in a previous video, those enteroendocrine cells. So the stomach can stimulate the stomach. And on the peptides, it can then vary how much time a meal spends in the stomach. So for example, if you were to eat a plate of pasta, that would be processed through your stomach rather quickly because the stomach doesn't do much with carbohydrate, as opposed to protein, which would spend longer in the stomach because the stomach can digest peptides. So the head can stimulate the stomach can stimulate the stomach. The small intestine then also has a role. The small intestine's main role is to inhibit the stomach. So when there is material in the small intestine is a very clear message for the stomach. Slow down. Remember, this isn't a race. We're not trying to get material from one end of the GI tract to the other as quickly as possible. Instead, we want to do this, take as much time as needed to get all of the nutrients and calories. And so when there's food in the small intestine tells the stomach to slow down, both through a neural reflex, where stretch in the small intestine can inhibit the motility and secretion of the stomach. This is known as the enterogastric reflex. But also the small intestine makes hormones such as CCK or cholecystokinin, secretin, and others like gastric inhibitory peptide. Just the very name tells you that this small intestine hormone will inhibit the stomach. And so there are a number of ways to control the stomach. There are a number of ways to control the liver, the gallbladder, etc. And this gets back to the small intestine. The small intestine is primarily for digestion, and most of its secretions are thus exocrine secretions. But it does make a number of hormones because, like other regions of the GI tract, it has enteroendocrine cells. And so therefore, as I had just mentioned, it makes a number of hormones which inhibit the stomach. But these same hormones, if they are signaling the presence of food in the small intestine, can then stimulate the pancreas, the liver, and gallbladder to release their secretions to help the small intestine with the digestion of this food, and can cause the dilation of the sphincter at the hepatopancreatic ampulla. Okay. And so the control of digestion was one of the topics here. Before talking about the process of digestion from start to finish, maybe just a few words about enzymes. Enzymes are proteins, while RNA can serve as an enzyme. That doesn't affect us here. And enzymes are produced by distinct parts of the GI tract. So there are enzymes produced in saliva. There are enzymes produced in the stomach, in the small intestine. And the liver makes many enzymes. The pancreas makes enzymes. The liver's enzymes are primarily for use in the liver, not secreted through the bile ducts, however. The liver makes bile, which is concentrated in the gallbladder. And so the process of digestion involves many players coming from different parts of the GI tract. Just a word on enzymes in general, and at the bottom of the playlist, I have a couple of videos on enzymes in general. One is, we need chemical reactions to occur to keep us alive. However, chemical reactions, while they can occur on their own, you can actually make glucose without any enzymes whatsoever. These reactions simply do not occur fast enough under natural conditions for us to maintain life. And so if we want certain molecules to form or certain molecules to be broken down, we have to make that happen. And so enzymes are proteins which cause chemical reactions to happen. The more official explanation is that they lower the activation energy so that a reaction can occur at a lower amount of energy than would otherwise be required. Why? Well, the analogy often used is that of a lock and a key, so that the enzyme has a spot called the active site, and the molecule that fits into this active site, therefore, is very specific to it. So just like your car key will only fit your car's lock, the active sites of specific enzymes are specific for different types of molecules. So trypsin has active sites which fit certain reactants, as would pepsin and other enzymes as well. Because proteins thus need to keep a specific shape, anything that changes the shape of a protein is potentially very bad for the body. So pH changes, they are bad for protein shape, all right? And so therefore, if you change a protein shape, it may no longer perform the function that it was designed to, and thus this is bad for the body. But other things like heavy metals, alcohol, high temperatures, etc., these can change an enzyme's shape. This would affect then the active site, and this would be bad for proteins in general, and then for these enzymes. Many enzymes work in cascades. So, starch is not digested in one step by one enzyme, but rather over a series of steps by a series of enzymes. Some of these actually then require cofactors as well. So these cofactors are called coenzymes. They do not make the chemical reaction occur, but they may bring something to the chemical reaction and thus help this chemical reaction. Many of the reactions of digestion are what are known as hydrolysis reactions, so that a water molecule is added and thus a big molecule is split into smaller ones. So in general, if this is the digestive tract, you have a problem. One, what you have eaten has big molecules like starch, animal proteins, plant fats, etc. This is simply too big to fit into the capillaries or into lymphatic vessels. So the fact that these molecules that you've ingested are too big to transport, that's the first problem. But then they belong to another species. What, in your body can you do with starch? Nothing, it's a plant protein. What can you do with chicken muscle? Even if you were building human muscle, you can't use chicken proteins in human muscle. You can't use corn oil for your adipose cells. So these molecules are just too big, plus they came from another species. You can't use them. All right, so that's a problem. But if you could break them into pieces, that changes everything. So if you could break these big molecules from another species to their building blocks, you could now reassemble the building blocks as you saw fit to make human molecules. This is not only, changing it, this is what's required for life. You need these molecules for calories, for the building blocks of your molecules. So the process of digestion is simply, you're going to break down the big molecules that you ingested in your diet into the small monomers that compose them, so that these can then be absorbed into the blood, or in the case of fat droplets, which I'll get to, into lymphatic capillaries known as lacteals. Now, to discuss this, we should take then each of the classes of molecules. Let me give a quick overview, but then I go through it step by step as well. Let's start off with, say, a carbohydrate like starch. Starch is too big. It's a polysaccharide, plus it comes from plants. However, the enzyme in saliva known as salivary amylase can break up starch into smaller molecules known as dextrins. So short chains of carbohydrates, of monosaccharides, are known as dextrins. So salivary amylase breaks starch is the reactant, into dextrins. Dextrins are then the product. Now, salivary amylase works in the mouth, but it then will denature. Watch the shape change. All right, so the shape changes when it hits the acid of the stomach. So acidity is important. Some enzymes only work in acid. Other enzymes can't work in acid, only work in basic solutions. So salivary amylase is inactivated in the stomach, unless, of course, we've eaten a big meal and a little bit of it has been pushed into the fundus of the stomach, in which case it hasn't mixed with acid yet, and salivary amylase could continue to work. All right. So salivary amylase stops working. The stomach really doesn't digest carbohydrates much. But once this material goes into the small intestine, pancreatic amylase, so the pancreas also makes amylase, this then can continue breaking starch into dextrins. The small intestine has, along its brush border, enzymes like dextrinase and others, which can then take short chains of dextrins and break them into individual monosaccharides of glucose. Glucose is an essential molecule for life. And so you've converted starch, which was simply too big, into glucose, which is small enough to be transported and essential for life. And so dextrinase in the small intestine breaks up dextrins. There are other molecules in the diet as well, such as disaccharides. Disaccharides can be split into their two monosaccharides by specific enzymes. So the small intestine makes the enzyme maltase, which breaks up the disaccharide maltose into two glucose units. The enzyme sucrase breaks up the disaccharide sucrose. The enzyme lactase breaks up the enzyme or the disaccharide lactose. And so then these disaccharides can then be absorbed and used for nutrients as well. That was the very quick overview, but in these videos, carbohydrate digestion 1, 2, and 3, I go over them into greater detail. Finally, a couple of thoughts on the large intestine. There shouldn't be much in the way of carbohydrates reaching the large intestine, so all of the starch should have been digested by that time, as in the disaccharides had absorbed. There are two exceptions. One is cellulose, what we call fiber. It's a polysaccharide. It's made of sugar, but it can't be digested because we don't produce cellulase. Cows do, termites do, but we don't. And so cellulose, while a polysaccharide, will pass through the large intestine as fiber. Now, it does have many benefits, like reducing weight gain, and that can be addressed separately, but it serves no nutritive function. If you were starving to death and ate cellulose, you would still starve to death. So it passes through unchanged because we don't have cellulase. Also, If someone is lactose intolerant, the small intestine has stopped making the enzyme lactase. So lactose was not broken up in the small intestine. It reaches the large intestine where the bacteria there do break it up. But the products of this, producing gas, might make someone uncomfortable to the point that they would attempt to limit the amount of lactose in their diet in the future. And so that is what is known as being lactose intolerant. You've stopped making lactase in the small intestine, and now lactose is broken up in the large intestine. Okay. Once again, far more in the individual videos, but that was a quick synopsis of how carbohydrates are digested. Also a quick synopsis of how proteins are digested. A protein is a chain of amino acids, all right? Amino acids have both an amino end and a carboxylic acid end, and so the peptide here, the protein, has an amino end and a carboxy end. Some enzymes are called carboxypeptidases. They can start at the carboxyl end of a protein and cut the peptides off one at a time, okay? Other enzymes are called aminopeptidases. They could do the same from the other direction, from the amino end, take off an amino acid from the amino end. And if you could do that, you might say, well, aren't we done? Because you could now break up the protein. The problem is proteins are just too big, all right? And so this isn't efficient if that's the only way that you're breaking up proteins. So what's important first is a set of enzymes known as endopeptidases. Say pepsin. Let's say pepsin is this purple enzyme made in the stomach. Pepsin then cuts in the middle of the chain, but typically only at specific amino acids, say the pink ones. And so after a protein passes through the stomach, instead of one big chain, you just have shorter chains because endopeptidases like pepsin cut in the middle. In the small intestine, trypsin, made in the pancreas, would be an endopeptidase, which would cut, say, at the yellow amino acids. Maybe chymotrypsin would cut at the blue amino acids, etc. And so what we see is that there are endopeptidases, which cut in the middle of chains at specific amino acids to make one long protein into a series of chains. And then from there, then the exopeptidases become more efficient in the small intestine. Now, I go through this in far more detail, talking about pepsin and all of these others. Pepsin is what's called an aspartate protease because its active site has aspartate amino acids, and therefore it can only fit certain amino acids from your diet. So the big amino acids, which have those phenyl rings, fit into that active site. And pepsin is secreted in an inactive form called pepsinogen, which is activated in the acid of the stomach. So pepsin is active in the stomach because it is acidic there. Now, the other endopeptidases, like trypsin, they are called serine proteases. They have a different active site with a different pocket, which includes the amino acid serine, making that active site. So it has a different shape, and thus different amino acids or different amino acid parts of the protein that you just ate will fit into this site rather than pepsin, which would fit different amino acids. And so serine, trypsin, trypsin, chymotrypsin, these are serine proteases. They are activated in a different way. And so what happens is that the small intestine has a brush border enzyme known as enterokinase. Enterokinase will activate trypsin by cutting off a specific sequence from one end of trypsin. So the pancreas makes trypsin in the inactive form of trypsinogen. This enterokinase activates it to become trypsin. And then trypsin then activates other enzymes. So enzymes are made in inactive forms so that they don't digest the cells which make them. And then they get activated in different ways. These pancreatic enzymes are activated then in the small intestine. Okay. And so the endopeptidases, they then have cut the large protein chains into smaller peptides, and now the exopeptidases can efficiently start at the carboxy end if it's carboxypeptidase, or the amino end if it's aminopeptidase, and then break these into individual amino acids. The pancreas makes carboxypeptidase. The small intestine makes aminopeptidase, which is a brush border enzyme. These now amino acids can be absorbed into the small intestinal cells and from there into the bloodstream. A couple of quick thoughts on the digestion of fats. First off, the most common lipid molecules in our diet are called triglycerides. Triglycerides are made by joining fatty acids to glycerol. Each glycerol can fit 3. And there are lots of different kinds of fatty acids, and that's why fats and oils vary from each other. Some fatty acids are short, medium, long, very long. Some have no double bonds and are saturated. Some have double bonds and are unsaturated or polyunsaturated. The hydrogens can be on the same side of the carbons in a double bond. That's a cis fat, as opposed to a trans fat where they're on opposite sides. See, these red dots are on opposite sides as opposed to being on the same side. And so because there are different kinds of fatty acids, then there are different kinds of fats and oils, because different fatty acids then have been joined to glycerol. If all of the fatty acids have straight chains and are saturated, that would tend to make a fat, which would be solid at room temperature. So if you took ones with no double bonds, that would make a fat. Whereas if you had unsaturated fatty acids, which then had a kink in the chain, that would be harder to pack into a solid at room temperature than you would have an oil. So these double bonds, which are unsaturated because you don't have as many hydrogens as you could fit 2 more hydrogens there, that then is an unsaturated triglyceride, which would tend to be an oil. So we ingest all of these different kinds of triglycerides, but then we break the fatty acids off with enzymes called lipases. So a lipase, could break off that fatty acid. So instead of a triglyceride, now you have a diglyceride. We could break off, another fatty acid to make a monoglyceride, et cetera. Now we have glycerol and 3 fatty acids. There are different kinds of lipases. So we have lipases in saliva, lingual lipase. We have a lipase in the stomach, very important for children who are using milk as part of their in their diets. And so there's gastric lipase, once again a bit more important in infants. But then the small intestine and then the pancreas make lipases as well. These tend to be require bile salts, and so some lipases are bile salt dependent. So there are a number of lipids in our diet, and we make lipases in a number of parts of our body: our mouth, our stomach, our pancreas, our small intestine, which break down lipids. The liver even makes hepatic lipase. There's more in our diet. We don't have to go into this, but we ingest a great deal of DNA and RNA. There could be 6 feet of DNA in a single cell, like a human cell. And so because you're ingesting lettuce DNA and banana DNA and chicken DNA, we obviously want to use that for food. So the small intestine makes a whole host of enzymes, like deoxyribonuclease, ribonuclease, phosphatases, which can break these molecules into their components as well. We absorb water, we absorb vitamins, we can absorb salts. But the big picture in digestion is this. You ingest molecules which are just too big. They're too big to transport in blood or lymph. Plus, they came from another species. So you can't use molecules which came, proteins from another species or fats from another species, etc. So you make enzymes along your GI tract. These enzymes break these big polymers into the smaller building blocks, the monomers. These are small enough to transport, and now you can reorganize them in the way that you choose to make human molecules. And so you break starch into monosaccharides like glucose. You break proteins into individual amino acids. These can be absorbed into the capillaries found in the small intestine, the blood capillaries. When you break up lipids, small chain fatty acids can also go into the blood. The large chain fatty acids, they are repackaged in intestinal cells to form fats, which are put into these lipid protein balls known as chylomicrons. Chylomicrons are shipped to a lymphatic capillary in the villi of the small intestines called lacteals. So this green tube is representing a lacteal, a lymphatic capillary. And when we ingest lipids, chylomicrons contain the lipid protein balls from those lipids, which instead of going into the blood capillary, then go into this lymphatic capillary. And so thus your body, which is very high in its energy needs and its nutrient needs, can then make it through the day because of the calories and the essential nutrients present in the blood and in the lymph resulting from the food that you ate.